Dear students, myself Suryaprakash, Associate Instructor in the course Fundamental of Genetics offered during second semester of BSc AG degree program. So in this continuation, we will discuss about the sex determination in plant as well as in animals as well as in plants. So students, if we consider the definition of genetics, that is the study of heredity and variations. This heredity refers to that the transmission of characters from parents to their offsprings. But in case of variation refers to the differences among the individuals for one or more characters. This variation is very much essential, is very much required, and we can say that this is the basis for any type of improvement, either in desired direction or for the evolutionary process. This variation is very much important. So when we talk about the creating a variation, so in that context, the sexual reproduction plays a greater role. It is the most effective methods of creating variations, and that can be utilized for the generation advancement. So in case of sexual reproduction, what we know that it is the fusion of two haploid specialized cells. That specialized cells is called as the gamete, and these gametes fuse together, and these gametes are produced in the reproductive organs of different sexes. That is called as the gonads in case of animals. So depends on the production of this gametes in the respective reproductive organs. The individuals may be either male or female. So we will discuss here all about this sex and their determinations. So what is the sex? It refers to the contrasting features among the individuals of a same species. Accordingly, with the presence of different sex organs in individuals, it may be either male or females. These two organs are present in different individuals in most of the animals, but there is certain examples where both the sex organs, either male or females, are present in the same individuals, like in case of earthworm. So in animal and plants as well, the male and female parts are very much essential to be utilized during the sexual reproductions. Now we consider the sex determinations. So it is the biological system of sex differentiations, the system of differentiating the sex of two, either male or females. It also determines the sexual characters present in their different respective individuals. It utilizes various genetical concepts for the development of male and female individuals. So why does sex exist? Why it is needed? So as I earlier told that it is required for creating the variation to participate in the sexual reproductions. For what purpose? This may be for the evolution benefit to the organism, and by adjusting themselves in changing environments. Second, case of sexual reproduction, this sex is only way to create the variations. So next, another is the combining beneficial mutations and removing deleterious mutation is also very much important for this crop improvement. So when we talk about the sex determination in animals, there are some historical hypotheses for this sex determinations. There are three basic historical hypotheses. That first one is that the origination of sperm. We know very well the sperm is originated in the testis. When the sperm is originated in right testis, it is supposed to produce a male baby, whereas if that sperm produced in left testis, it is producing the female babies. The second hypothesis, what the historical importance is the parental strength. If the father is stronger than the mother, then in that case the chances of male baby is more as compared to when the mother is stronger than the father. In that case, female baby will be produced. Third hypothesis is the direction of wind during coupling. So it is assumed that if there is northerly winds at the time of coupling northerly wind is there. So in that case, the male baby will be produced, whereas the southerly wind, if southerly wind is going on, then in that case the female baby will be produced. But we have to much more concentrate on the experimental evidence. There are some of the experimental evidence for the sex determination. The first and foremost thing is the discovery of 6 chromosome, that is the X chromosome, by Hanking in the year 1891. Second, the case in history is the discovery of supernumerary chromosome by McClung in the year 1902, in case of while he was dealing with male grasshopper, and Wilson and Stevens in the year 1905 when he studied the proteinor. And next, the discovery of Y chromosome was done by the Stevens when he studied the Drosophila in the year 1908. So when you talk about the sexes, there are two types of sex characters, that primary sex characters and secondary sex character. Primary sex characters are generally related with the gamete producing organs, that is gonads, and its development depends upon the presence of gene in zygotes. Whereas in case secondary sex characters, it shows the consistence difference among female and male. There is consistency, there is much more consistency in case of secondary sex characters. It includes the genetical ducts, the facial hairs, the mammary gland, and also the pitch of voices. Generally we see the pitch of voices is also differ with the character with the sexes, that the female the pitches of voice is generally is melody than the male individuals. Development is of this secondary sex character is induced by the hormones, and their intensity is modified sometimes by the external administration of the hormones. These hormones, if administered, then there is chances to develop the secondary sex characters in an individual. So gonad differentiation in animals plays a very important role, and here HY antigen plays a key role in this differentiations. If there is XX or XY zygotes, then in that case, if there is presence of HY antigens, the cortex regresses and testis is developed. Here the medulla is at center and cortex is present at the peripheral region of undivided zygotes. So if there is no HY antigen present in that case, so the medulla regresses and develop into a ovary. So the sex determination, there are different mechanisms of this sex determination. That first one is the environmental, then chromosomal, then genic balance. There are three different ways, mechanisms of sex determinations. So when we talk about the environmental sex determinations, in that case zygotes do not differ themselves genetically for their sex differentiations. This sexual differentiation is only determined by the environmental factors. If we consider the example of environmental sex determination, the first and foremost example is the larvae of sea worm, that is Bonilia, when attached with the proboscis of female insects, that develops into a male, whereas those individuals, those larvae which are free, which are not attached with the proboscis of the female insects, they are free and then they are developing in the females. So this is assumed that this is due to the production of hormones in proboscis by the female worm, female insects. Second example is the sea worm Dinophilus, where larger size eggs develop into the female, whereas the smaller size of eggs develops into the females. The third one example is the sea fishes labroids, where the social orders of individuals in a colony determining in the sex. Basically in this, but in one colony there is only one females, and then the other one is depending upon their the residential order, they differentiated in the different sexes. Second one is the chromosomal system of sex determination. In this case, individuals differs with respect to the number or morphology of homologous of one chromosome pair. That chromosome is considered as the sex chromosome and also called as allosomes. Chromosomes do not differ in number and morphology. Those chromosomes which do not differ in their morphology or in their number are called as autosomes. But this is not common for all animals. There are examples where two types of sex chromosome, that X chromosome and Y chromosome, is observed in different animals. So the mechanism of chromosomal sex determination is grouped under 5 different types, that XX producing the female, whereas XO male, XO female and XX male. So in this way there are 5 group are there for the chromosomal system of sex determination. We will discuss one by one to all these systems. So when XX producing the female and XO producing the male systems, here this system was found in the insects belong to the Orthoptera family, and for the example of this is a grasshopper and proteinor. In this case, females have two X chromosome, and that's why it is considered as, it is called as the homogametic sex, while male have only one X chromosome. That's why it is called as the heterogametic sex. In case of grasshopper, we can see here that the gametes is producing from the one X only from the female parents, and one X is producing only from the X, whereas the other one is the null gametes. So when combining these, the progenies are produced with the double X by the female, and that XO is the male. The next system is just reverse to this earlier system, that when XO producing female and XX male producing male. So this type of sex determination is observed in the insect Phumia, where females have only one X chromosome. That's why it is called, it is considered as the heterogametic sex, while the male have two X chromosome and referred as the homogametic sex. We can see here the parents of parents of XO female and male, when combined together, they form the gametes, and the progeny were developed in a 50% 1:1 ratio. Third system is the XX female and XY male. This system is much more common in human, in mice, Drosophila, some of the fishes, and some of the amphibians. This system is generally common. So in this system, females have two X chromosome. That's why it is referred as the homogametic sex, whereas the male have one X and one Y chromosome and referred as the heterogametic sex. Here we can say that in case of human, when female is crossed with the male XY, in case of female, one gametes X is producing, whereas in case of male, two gametes, one X and other one Y is producing. When these are all combined together, the progeny is produced in 1:1 ratio. Here one mistake is that it is XY. So next system is the XY female, just refers to this earlier one. This here XY producing the female system, female individuals, and the XX producing the male individuals. Here this type of sex determination is observed in birds, in reptiles, and silkworms. Then here the females have one X and Y chromosomes. That's why it refers as the heterogametic sex, while males have two X chromosome and referred as the homogametic sex. The parents are combined together and produce the progeny of male and female in 1:1 ratio. Another system of sex determination is their diploid levels. When individuals have diploid level of ploidy, it produces the female, whereas if it is haploid, then it is supposed to produce the male systems, male individuals. So this system is generally observed in the family Hymenoptera. The example of this Hymenoptera insects are honeybee, ants, and termites. Here somatic chromosome of female is diploid and male is haploid. This system was first demonstrated by D. John in the year 1850. Here we can say that queen has n haploid eggs and drone has haploid n sperms. So if there is no fertilization takes place, then that haploid larvae producing the drones. Whereas when there is fertilization of the queen, the eggs of queen and drones, as a drones, queens, eggs of queens and sperms of drone, when fertilized together, it produces the zygotes having diploid conditions. So these diploid larvae, when fed with the royal jelly, so in that case they produce the queen, which are female, fertile females. But if that diploid larvae is not fed with the royal jelly, so in that case they produces the sterile females, and generally we call it as the workers. So next system of this sex determination is the Genic balance system, where the sex of an individual is determined by the balance between genes of male and those for the female. So this system was first discovered by the Bridges in the year 1916, where he found the X and Y were producing the females, whereas males has only XO chromosomes in case of Drosophila, while studying with the inheritance of vermilion eye. So sex here in this system, in this Genic balance system of sex determination, we have to estimate the sex index, which is the number of X chromosome, that of the number of autosomes divided by the number of autosomes. So if there is equal set of autosomes to that of the X chromosome, it is assumed to develop into a female individuals, whereas if half of the X chromosome to that of the autosomes, it assumes to develop a male individuals. So this is about the Genic balance system, where the balance would be there. One thing, gynandromorphs, it is produced in Drosophila, where the parts of the body showed male phenotype as well as the female phenotype simultaneously. Male phenotypes is extended up to the half of the body. Sometimes it is extended up to half of the body. In some flies, male and female parts runs longitudinally, whereas in some cases it runs transversely. It means the female and transversely, and in case of longitudinally, the female parts and the male parts are just divided longitudinally, whereas in some insects of this Drosophila, it is transversely divided, the male and female parts. So gynandrophore arises from the X zygotes having XX chromosome, that is due to this type of system, this type of you can say the individuals is developed due to the irregular distribution of X chromosome during different phases of cell divisions. The extent of male parts depends on the stage of embryonic developments, then in which stage this irregularities of X chromosome takes place. Now if we can now next one, that the sex determination in plants. This all about the sex determination in animals. Now we consider we can discuss about the sex determination in plants, which is very much complex mechanism that involve various factors. In case of plants, we know the sex are either in same flower or in different flowers. So accordingly, there are monoecious plants and dioecious plants and hermaphrodite flowers as well. So in this case, sex determination is very much difficult. So if we talk about the crop improvement, a crop to be used in a breeding program, it determines the sex in seedling stage, that reduces the time for for sex determination as well as the input cost we can reduce, we can minimize. Morphologically, markers like flower type, etc. for conferring sex of the plants also requires tedious work and the larger area to select sufficient number of plants, and for this, therefore, here we can determine the sex by molecular markers that play important role in determining the sex in early stage of the plants. So when we talk about the sex determination in plants, it is in this most plants have both sexes on each plant, and some plants have one sexes per plant, like in case of bryophytes have separate sexes in their gametophyte stages. So the presence of male and female parts in this, the mode of sexuality differs in the flowers. So accordingly, it may be classified in 8 different types, that hermaphrodites, that all perfect flowers are present. In monoecious, in case of monoecious, separate females and males flowers are there, but they placed on the same plant. In case of dioecious conditions expression, the separate female and male flowers are present on 2 different plants. There is next expression is the andromonoecious, where the perfect flowers and male flowers are present on the same plant. Gynomonoecious, in that case, the perfect flower and female flowers present on the same plants. And trimonoecious, similarly here, the perfect flower, female and male flowers present on the same plants. And there are 2 more, the androdioecious and gynodioecious are the expression of sex, where the perfect flowers and male flowers on separate plants, and perfect flower on and female plants and female flowers present on the 2 different plants. So similarly, as in case of and sex determination mechanism in case of animals, in this plant it is same, the environmental mechanism, the chromosomal mechanism, and the genic mechanism. So we can deal one by one here. So the environmental sex determination in the plant, it is due to the environment, or it is greatly affected by the environments. So for example, in case of equisetum, optimum conditions is assumed to produce the female individuals, whereas if it is faced by the adverse conditions, it is assumed to produce the male. Some of the nutrient doses and some other things are also responsible for the production of male and female individuals. In case of cucumber, melons and cannabis, the sex of flower is affected by some of the environmental factors like nutrient doses, etc. Sometimes temperature, day length, and gibberellic acid, use of gibberellic acid also will play a greater role in the determination of sex, in the production of promotion of different sex individuals. In cannabis, the gibberellic acid induces the development of only the female flowers. The chromosomal system of sex determination is also there in case of plant, where XX produces female and XY produces the male individuals. Here X and Y chromosomes are identical in their morphology and they segregate randomly. This system is found in spinach and asparagus. In addition to the major gene affecting sex, the Y chromosome thought to contain a gene that suppresses the carpel development and another gene that promotes the stamen development. Mutation in these genes leads to hermaphrodite flowers, hermaphrodite individuals, and asexual flowers. Both of these types are known in asparagus. The next system is the X and Y producing the female, whereas the male have XX chromosomes. This system of sex determination is found in Fragera, Elytria, wild strawberry, unisexual, and this may be either tetraploid, hexaploid, or octaploids. Here the female produces two types of gametes with respect to sex chromosome. That's why it is referred as the heterogametic sex, and male produces only one type of gametes, sex chromosomes. So that's why it is homogametic. Heteromorphic sex chromosomes, where X and Y chromosomes are distinct in their morphology. Generally, they are unable to pair over a significant portion of their length. So this way the morphologically differences are present. That's why it is a heteromorphic system of sex chromosomes. So here the female has XX chromosome, whereas the male has XY chromosomes, and found in cannabis. Here egg cells have one X chromosome, half of the pollen grains have one X, and remaining half of the chromosomes have Y chromosomes. Individuals have Y chromosomes. Random union taken place among these gametes, producing 50% female having XX chromosome, and 50% progenies are male which are having XY chromosomes. This operates in the same manner as that in case of the mammal. So organization of X and Y chromosome is easily observed in Melandrium, where we can see here that the four parts present in the Y chromosomes. That first and topmost part is the female suppression regions. Here, if this portion is deleted, then it is developed into the female, whereas the male promoter regions, if this system is deleted from the Y chromosome, this portion is deleted from the Y chromosomes, then the female baby will be produced. The other one is the pairing regions. There are pairing regions where the X and Y chromosomes generally pair with each other. Next system is the X1, X1, X2, X2 producing the female, whereas the X1, X2, Y1, Y2 producing the males, and it is found in humulus lupulus. Here eggs have the chromosome constitution is X1, X2, whereas the chromosome constitution of male has X1, X2 and Y1, Y2 in their pollen grains. When these two male and female parents comes together, they produces the different gametes, and then they fertilize and the progenies is developed as X1, X2, Y1, Y2 as male, whereas X1, X2 and X1, X2 as female. So next system is the genic balance system of sex determination. This is found both in case of monoecious plants as well as in case of dioecious plants. Here the sex of individual is governed by the genes present in the individuals. Here single locus that has a single gene plays a major role in the determination of sexes. Here sometimes multiple loci is also responsible for this purpose. So where two or more genes are responsible for the sex determinations. This system is operating in case of papaya, in case of maize, in case of spinach. The produces female plants, whereas capital M1 and small m produces the male plants. Capital M2 and small m produces the hermaphrodites. And if the genotypes having capital M1 and capital M1 and a small m2 are invariable. That is, the M1 and M2 alleles are recessive and lethals. So this way we can see here the sex determination in papaya. Developmental steps that affected by the sex determination in case of maize, we can see here the different parts of the tassels and ears are there. So these are the 6 organs of the maize plant, and the sexual development of male and female florets are here. The maize inflorescence are there. So if we see the differences between the genetic and environmental sex determination, so in case of genetic sex determination, the sexes segregate developmentally during the meiotic stage, different meiotic stages during segregation, and whereas in case of environmental sex determination, the segregation is due to the external regulation of gene, that may be due to the nutrient, may be due to the hormones and some other means. In case of genetic sex determination, the expression patterns are invariable, whereas the gene content variable. Whereas in case of environmental sex determinations, the gene content is mostly invariable, whereas the expression pattern is varied with the individuals to individuals. So thank you very much.